Yes, go on, please. Yeah, go on, go on. Okay. So basically, one second. Okay, let's go back to your gene libraries. Okay. So see, basically, what is the idea of a gene library? See, what we are going to do in a gene library. See, we need to keep all your genes with us. Okay. So we need to keep it somehow or the other. So how are we going to keep it? We are going to keep this as clones. Okay, but now even if we are keeping it as clones, like we need to have some procedure. We need to have, like, either we are going to have only the gene or only the DNA which codes for the gene, or we are going to have fragments of the DNA. Okay, so we have two different ways in which we can do this thing. So the first way is by having your genomic library. Okay, so genomic library means you are going to have the entire genome. Okay, see, the idea behind this is, in genomic library, you are going to have the entire genome like this. Now, in this genome, there are going to be several genes which are going to be present. Okay, so let's say one gene is present out over here, one gene is present out over here, one gene is here, and one gene is here. So in this thing, what is our idea? We are going to make use of your restriction endonucleases, and we are going to cut these fragments off. Okay, now we don't know, like, exactly how big is this fragment, how is this fragment, we don't know anything. So ultimately, what are you going to get from here? You are going to get that, okay, chalo, this is one fragment, this is another fragment, this is another fragment, this is another fragment. Now you don't know which fragment contains which gene. Okay, because at this moment we are not going to be capable enough of knowing it. Okay, but now what you can do is, see, what you can do, you can take these fragments, okay, each of these fragments. Okay, so let's say this is for insulin, let's say this is for your amylase, let's say this is for, let's say, Tylen. Okay, let's say this is going to be for your pepsin. Okay, so let's imagine, we have these genes present. Now I don't know, like, at this moment when I take this thing in a test tube and when I cut this thing, I don't know which fragment is going to produce what. Okay, so now what we do, guys, we are going to take each of these fragments. If we take each of these fragments and we are going to put it into your vector, that is your plasmid. So see, each of them will be put into your plasmid. And now once I have put each of them into a plasmid, each one of them will go into another organism. So it will go into this. Let's say it will go into this one, will go into this one will go into this, like this, and this one will go into another one like this. Now what are we going to do, guys? See, when we have these bacteria, they are going to be transformed. Now I'm just going to look at this thing, I'm just going to look at this, because they are going to keep on multiplying. So one, this is going to convert into two, two are going to get converted into four, and four into eight, and so on and so forth. This is going to happen. But now, guys, what did I tell you? Whenever the first one with the original plasmid with this gene is going to be present, each and every cell out over here is going to receive the same gene, right? The plasmid is going to be replicated and it is going to be given to each of them. So now, guys, what happens? When I look for these, I screen them. I screen them, meaning I check for what activity. So I will check if this is giving me insulin activity. If it is giving me insulin activity, what does this mean? That this fragment, it contained insulin. Same way, I am going to take this one and now I'm going to check for Tylen activity. So if this produces Tylen, now what I can say, yes, this fragment contains Tylen. I will check this thing for amylase activity. So amylase, yes, then this is going to be, yes, this is also going to be maybe your pancreatic amylase. Let's imagine. So yes, it gives me that pancreatic amylase activity. And this pepsin, so just, let's see, we check this thing for pepsin activity. So immediately this is going to give you pepsin activity. And this is how you'll have to keep on checking for each fragment, because here we don't know which fragment is going to be carrying which gene. Okay, everybody all right with this thing? Clear with this? Guys, quickly, just tell me yes or no. Okay. So this is our genomic library. Okay, but genomic, after your genomic library, guys, we are going to have your cDNA library. cDNA library is what? It is your complementary DNA library. Okay. Okay. Now, see, in your complementary DNA, now see, what is the problem with your eukaryotes? Okay. So, in your eukaryotes, see, the main problem is you are going to have a DNA. Okay. So, this is your DNA. DNA is going to give rise to your mRNA. Okay. And mRNA is then going to give you your protein. So, this is how it works. Right, this is how it generally is. But now when we talk about this DNA, on this DNA there are going to be several regions. So we have the coding region, we have the non-coding region, then once again we have the coding region, then once again we have the non-coding region, and this thing keeps on repeating itself: coding, non-coding, coding, non-coding, coding, non-coding, coding, non-coding. And because of this, now what happens? See, when this one, okay, when this DNA is going to be transcribed, it gives us your mRNA. But this mRNA, guys, is it really going to be immediately used? No, 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 no. This is not going to be used. It has to undergo post-transcriptional modification to be fully functional mRNA. So now when we get this mRNA, guys, initially, what are you going to see? You have exon, intron, exon, intron, exon, intron, exon, intron, right? And out of this, who are the coding regions of this mRNA? So we are going to have exons, 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 exons. These are the coding regions. Whereas introns, these are actually a waste, basically. Okay, they are not the real coding regions. Okay, so now let's try to see. So guys, after your post-transcriptional modification, okay, so what is post-transcriptional modification? What are you going to see out over there? You're going to see capping, then you're going to see splicing, you're going to see tailing, right? I'm sure you guys have done this thing. Okay, chalo. So now what happens, guys? See, you are going to see that during this splicing, you are going to see that these introns, they are going to be removed out. Introns are all thrown out. Okay, because introns were not coding only. That is why. So when we talk about your fully modified mRNA, we are going to see that it just has your exons. Now, what does this mean? Okay, if this is the one which is having exons, so only the coding regions were actually, important. The non-coding regions were not important, so they will be actually taking up space. So if I take this entire DNA and I put it into your plasmid and then into your E. coli, maybe this will not be read at all, because the bacteria may not have this splicing mechanism, it may not have this capping mechanism, it may not have this, the tailing mechanism, right? So this will be a waste. I cannot use this thing. So now what do I do? See, I have got mRNA. mRNA is completely filled with this exons, exon. This is your completely modified mRNA. Now what do I do, guys? I am going to add a reverse transcriptase here. Oh, just a moment. Okay, so now, guys, why are we using this reverse transcriptase? See, because reverse transcript, what is the idea behind this? This is the one who converts mRNA back into your DNA, right? But now, guys, at this moment, if we add reverse transcriptase, see what are we going to get? This mRNA, this is now going to get converted into your DNA. But this time, guys, what you are going to see, how is the change? Everything is going to be the coding regions only. See, here it was consisting of your coding, non-coding regions. Now here, when you add this thing, the non-coding regions are not going to be present. You are going to get only the coding regions. And guys, isn't this going to be good? Because you have removed all these waste or junk DNA, right? Now what we can do? Yes, now you're going to take this thing and you are going to put it into your plasmid. So this goes into your plasmid. And now the moment you have it in your plasmid, guys, now what? Now you take this thing, put it into your bacteria, goes into your bacteria. And now the moment it goes into the bacteria, now you're going to see that this bacteria is going to start multiplying. As it starts multiplying, guys, once again, what did we say? This is going to produce your insulin. Insulin, or whatever whatever this gene was present. Okay, everybody got this thing, guys? Guys, now clear? Those who had any issues? Guys, is everybody clear? Okay, any doubts, anything else? And guys, for humans, okay, for humans and everything, we will always go for your complementary DNA only. Okay, this is the most important thing. Okay, we won't go for this one. Okay, chalo. Then I had shown you this entire this thing also. Okay, chalo. Now, guys, let's try to move ahead. Let's try to see. Okay, now I told you from here onwards, we are going to have a little on the simpler part, general types of things. Okay, so let's try to see the next part now. Okay, just a moment. So now there are some photos and all which I have I have taken it from your textbooks only. Okay. 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 Now the next thing is, guys, we are talking about your biotechnology, but what are the different fields of biotechnology? Okay, so let me just quickly put it out over here. Sorry, what is happening? Okay, sure. So guys, these are the various fields that we generally come across, okay, with regards to your biotechnology. So let us just once again see what are the different fields. Okay, sure. Take it. Okay, so now guys, the first thing that we are going to have is your plant technology, plant biotechnology. Now guys, what does this mean? Now see, in plant technology, guys, what are we going to do? We are going to purposely change the genetics of the plant. Now why? Maybe you want to give it pest resistance, or maybe you want to give it some kind of your drought resistance or high salt resistance, or maybe more amounts of nitrogen fixation, right? So all these kinds of different genes that we will incorporate into the plant, that is going to be your plant biotechnology. Now always remember, guys, when we use your modified genes and all, now we call them as GMOs. Okay, always remember, see, GM crops or these are known as GMOs. Okay, what is the full form of GMO? Genetically modified organism. A genetically modified organism. That is GMOs. Okay, and now, guys, what is this GMO going to do? This GMO is going to give you all the desired characteristics. Maybe it is going to give you higher yields. Maybe it is going to give you better flowers. Maybe it is going to give you better stress resistance, dot resistance, all those things, guys. Okay, for example, if you remember, we had talked about BT cotton, BT cotton, BT brinjal, BT bhendi, right? So all those things, we were incorporating that Bt toxin gene into your plant. So when we were doing this, what is that? That is nothing but your plant biotechnology. Okay, along with that, guys, you must have heard of golden rice. Okay, which is going to have increased amounts of vitamin A, so that we can take care of the vitamin A nutritional deficiencies in people masses. So that way, so that includes your plant biotechnology. Okay, but now, guys, next one that we have industrial biotechnology. Okay, so in industrial biotechnology, guys, what are we going to do? We are going to make use of enzymes, like how we are going to produce enzymes, how we are going to produce different kinds of your maybe antibiotics, maybe what are the secondary metabolites, like your vitamins, your organic acids, your antibiotics, all those things. So basically, we are going to determine how the parameters and all work for that. So that is nothing but protein engineering and metabolic engineering. And the same thing, when combined together, we have a field which is known as metabolomics. Okay, so always remember this. Okay, so what is metabolomics, guys? Metabolomics is nothing but when you are going to work on all the different types of your metabolites. Okay, so when you work on all the different kinds of your metabolites and you get them from the organisms, that is nothing but industrial microbiology. Guys, once again, in your industrial microbiology, you're also going to have your this thing like beer production, wine production. Okay, so we have spoken of this thing in microbes in human welfare. So all that thing is included in industrial microbiology. Okay, the next field that is there in biotechnology is nothing but genomics. Oh, okay, genomics. Now, guys, what are we going to do in genomics now? See, there are two fields, okay, bioinformatics and genomics. They always go hand in hand. So, bioinformatics, proteomics, and genomics. See, this is that entire group. So what we do in genomics, guys, in genomics, the first and the most important thing that is done is, let's say this is your cell, inside your cell, this is your chromatin fiber, meaning basically this is your DNA. You are going to remove this DNA, and guys, this DNA is going to be read. Like, what is the sequence? How it is going to be A T G C C A T T G G C C C, something like this. How do we come to know that, oh, the sequence is like this? So for this, guys, we have a scanner, and that scanner is nothing but DNA sequencer. Okay, so that oh, okay, so that DNA sequencer is going to read this thing, what is present on this thing, guys. So that is going to give you that entire genome sequencing. Okay, guys, remember we had this human genome project. In human genome project, we sequenced our entire DNA. So we got all the, the various kinds of, the sequences, okay, for different kinds of genes and all on our DNA, guys. Okay, so along with that, we have also, sequence other organisms as well. Like we have Drosophila, we have E. coli, okay, musculus. Okay, so all those organisms have also been sequenced. Okay, so now, guys, what happens is, and this, guys, once we have this DNA sequencer, it is going to scan, and then this is going to give you this entire sequence. Now, guys, with this sequence, see, I told you what are the three fields: proteomics, genomics, and bioinformatics. So now, guys, once we have got this sequence, now from this sequence, we can, just put it into our computer. And in computer, we have different programs, like we have BLAST, we have FASTA. Okay, so basically these are some, this thing. So when you take this DNA sequence, let's say the DNA sequence look like this. Now the computer program, guys, is going to recognize what is the meaning of this thing, and it is going to give us, oh, and from this position to this position there is this gene, or from this position to this position there is another gene, and it will give you a complete analysis of it. That is nothing but bioinformatics. So with the help of this tool, guys, this blast fasta, then you are going to have comparison between different kinds of your genes and all. You can get exact information about what is happening inside the gene, what is happening with your DNA, that way. And then you can also do these things in this thing, like, you have got two different organisms. You can remove the DNA from both of them, and you can compare how closely they are related, or how widely they are separated. So that way, so all this thing is, guys, nothing but bioinformatics. Same way we are going to find out, see, gene functions, what is one gene going to do. Then you can also go back and you can just verify your pedigrees and all. So that is reverse genetics. So basically this field is your genomics. Okay. Then next thing, we have microbial biotechnology. Okay, so when we talk about microbial biotechnology, guys, our focus is mainly intended on use of your microorganisms for different kinds of products. So see, we are going to see microbial genomics. Sorry. Okay, so now in your microbial technology, once again, guys, so you are going to talk about all the genetics of microorganisms, how are they going to be, what is the DNA sequences, and all that studies. Okay, so microbial genomics. Then it helps in the production of your biofertilizers, maybe biofertilize, your biopesticides, and so on and so forth. So that way. So guys, when once again in your microbial biotechnology, we're going to look at your microorganisms and use of microorganisms for the production of various products. Okay, then after that, we have, see, basically when we talk about this IPM is nothing but integrated pest management. Okay, so that is nothing but when you're using biopesticides only, so that is the same thing, integrated pest management. Okay. So that is this thing. Okay, guys, then the next thing is we are going to have environmental biotechnology. Okay, now guys, today this environmental biotechnology, this field is like booming like anything. Okay, because for everything, guys, we are trying to find out different kinds of solutions. See, for example, like for most of the times what happens is big ships containing oil. Okay, sometimes they crash into one another, or then there is oil spillage and all. So people are working, guys, people are continuously working to remove that oil from the surface of the sea, because otherwise what is going to happen, then it is going to just cut off the oxygen from the atmosphere, and all the flora and fauna, whatever that is below it, it's going to die off. So now things like this, guys, finding out solution to this can be done with the help of microorganisms and all. And that is nothing but, we say, that is pollution control. Okay, this is just one type of thing I'm talking about. Then another thing, guys, is biofuels. So here you are going to see that with the help of microorganisms, you can create some methanol component. Okay, and that methanol or ethanol can be mixed with your fuels, and then it can be used in your cars and all. So that is nothing but your biofuels. Okay, then we have bioremediation, like taking care of the environment, making it once again proper and all. That is bioremediation. And also taking care of biodiversity, increasing the diversity of organisms in the entire environment. So that is environmental biotechnology. And last one out over here, guys, we have animal biotechnology. So in animal biotechnology, see, biotechnology, animal technology, they go work, they always go hand in hand. So what do we have out here? Yeah. So we are going to have this tissue culture. We are going to have this different types of your medicine formations, like obtaining different kinds of compounds from your bacteria meaning your animals out over here in this case. So immunotechnology, making use of your immunology, antigen, antibody reactions, and all, producing your hybridomas or producing your monoclonal antibodies, all that thing. Genetically modified organisms for increased yield, increased production. Then pharmacogenomics, meaning using your genomics, basically your idea, the entire idea about your DNA, and then producing your pharmaceutical ingredients so that they will affect only certain portions of your body. So that way. So then this, and then finally we have this gene therapy. Guys, in this gene therapy, what we try to do is, if by any chance you are having a defective gene, we are going to cut off that gene and we are going to replace it with the proper gene. Or by some or the other way, we are going to take a proper gene and we are going to insert it into a cell so that it incorporates it into the cell and produces the product that way. So all this thing is animal biotechnology. Okay, so guys, this is what we all need to know. What are the different fields of biotechnology? Okay, chalo. But now let's try to see. Okay, we are seeing that. Okay, chalo, we are having biotechnology. But guys, what are the biotechnological products that we see in our day-to-day life? Is there any such products out over there? Are there any recombinant DNA proteins or recombinant DNA vaccines? Anything that is available? The answer is yes, guys. Yes, there are various kinds of these things which are present. So let's try to see a small table in which we have different recombinant products that are produced by using rDNA technology, and what are they going to be used for. Okay, so let's try to see. Okay, so now guys, the first one we have anemia. Now, basically in anemia, what is going to happen? Your RBC count is going to go down low, the hemoglobin count is go down low, right? And because of that, deficiency of iron and all ultimately results into anemia. So guys, to treat this thing, what are we going to use? We are going to use erythropoietin. Because see, whenever you're going to use erythropoietin, guys, what is this going to stimulate? It is going to stimulate your RBC production. Okay, and immediately you are going to be cured of it. Okay, the same way, guys, we have your asthma. In your asthma, we are going to use interleukin 1 receptor. Okay, these are different kinds of receptors that are present. Okay, so interleukin IL-1 is one of them. Next, atherosclerosis. Now guys, in order to treat this thing, we are going to use platelet derived growth factor. Okay, at the time of parturition, so that the baby comes out easily, we are going to administer a relaxin. Okay, if by any chance you are having blood clots, to dissolve the blood clots, guys, okay, to dissolve the blood clots, we are going to make use of tissue plasminogen activator or urokinase. Okay, now what is this going to do? This TPA, guys, always remember, this dissolves blood clots. Okay, that is the idea. And guys, just like how we have this, okay, urokinase, there is also one more thing that is available. We call this thing as streptokinase. In your exams, guys, you are always going to get this question: what is streptokinase used for? Really, really simple, straightforward. It is used to dissolve blood clots in the body. Okay. Then, guys, the next thing we have interferons, okay, to treat cancer. Okay, so we have interferon, we have tumor necrosis factors. So tumor necrosis factor, we call this thing as TNF. Interleukins, there are different classes of interleukins which are going to help you, ward off cancer. Then we have macrophage activating factor, MAF. This is going to initiate that macrophages come and show phagocytosis that way. Now, interferons, guys, what are these interferons? I'll just tell you. Let's say, we have our body, okay, and in our body, let's say we have these cells which are like, housed together. Okay, so let's say these are our cells. Okay, so we are going to see a proper nucleus. Let's say in one of the cells, you are going to see that there is a virus attack, and because of this virus attack, this cell, it has fallen prey to it. Okay, but now what is going to happen, guys, when this one falls prey to that virus, before dying or before the virus completely captures this thing, it releases certain products. And what are these products, guys? These products, they are going to alert the neighboring cells. Kyori, there is a virus attack out over here. There's a virus attack. Be careful, be prepared, prepared. So basically, guys, this is like an alarm, 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 alarm. Okay, so that way. So what is this cell going to do? The moment this gets infected, it raises an alarm. And because this cell raises alarm, all the neighboring cells, they are going to be now completely aware that, oh, this kind of a situation is happening out over here. Now, what is this alarm? This alarm is nothing but interferons. Okay, these interferons, they help the other cells to recognize that something is wrong out over here and take appropriate measures. So that, okay. Then we have tumor necrosis factor. This is going to bring about necrosis. Necrosis meaning what? Death. Okay, necrosis is cell death basically. Okay. Then we have interleukins. As I told you, there are going to be different classes of interleukin. They are specialized for doing various jobs. Then we have macrophage activating factor, where it is going to initiate your macrophages to come and eat away the unwanted stuff. Okay, so for your diabetes, we have your insulin, once again, genetically engineered insulin. For emphysema, we have alpha 1 antitrypsin. Guys, this question is very important. Okay, they always ask you what is used for emphysema. Okay, hemophilia A and hemophilia B. This is always asked in your exams. Okay, so just keep this thing in mind. How do I remember this thing? I'll tell you. See, we have hemophilia A, hemophilia B. Okay, you just need to remember there are two factors, either factor 8 or factor 9. You go in chronological order. Hemophilia A, so then it is going to be factor 8, hemophilia, and 9, hemophilia B, it is going to be your factor 9. So chronological order. I remember it this way. Okay, then we have hepatitis B. For hepatitis B, we produce hepatitis B vaccine. Same way, guys, we have your rabies also. Okay, so for rabies, we produce a rabies vaccine. Okay, so these are, guys, some of the biotechnological products that we need to remember. Okay, but now, guys, apart from this, there is one more field in which we are going to make use of biotechnology, and that is nothing but healthcare biotechnology. So basically, that is nothing but production of your hormones and all. Okay, so let's try to see how it is going to be like. Okay, so now, guys, in the next thing that we are going to talk about is one of the most important questions. They always ask you the structure of human insulin, or how it is, what happens out over there. So that is what you need to know. Okay, but now let's try to see this part. Okay, so now, guys, first thing that we're going to talk about is your human insulin, or what we call this thing as humulin. Okay, humulin, or this is nothing but human insulin. We'll also see how we are going to produce this thing, like what happens. So first, guys, let me just show you how is your normal insulin, what really happens out over there. Okay, so, guys, see, when we talk about your insulin, guys, we are going to see that there are going to be three chains present, A, B, and C. Okay. So this is how the arrangement is going to be. But let me just once again show you how it is going to be like. Okay. Chalo. So guys, basically the idea is going to be this way. Okay. So now let's say you have first one, guys, you have this B chain. Okay. This is our B chain. Okay. Now at the end of the B chain, guys, one thing you always need to remember, we have your NH2. The B chain always ends in NH2. Okay, fine. Now this B chain, it goes for a little distance and then ultimately it is going to meet with your C peptide, or we say it is the C chain, if whatever you like to see. Okay. So then you, right? So you are going to have this C peptide. Okay. Okay. Now what is going to happen? See, after this C peptide, now you're going to see that, okay, this is going to be attached to your A chain like this. Okay, so something of this sort. So guys, this is how the idea is. Okay, but now. Okay, what we are going to see is, guys, that this B chain and A chain, they are going to be connected together. Now, how are they connected together? So you will always see that this B chain and A chain, they are going to have somewhat disulfide linkages, something like this. Okay. And these linkages, they are going to hold the two chains together properly that way. Okay. Now what happens, guys? See, this whatever insulin that we are talking about, again, same way you can see there are like, disulfide linkages within here as well. Okay. Now, in order to make this thing active, guys, see, this is your inactive. Okay. This is your pro insulin that we can call it. Okay. Pre-pro insulin. That is what we call this thing as. Okay. So let me just show you. This is pre-pro insulin. Okay. And this pre-pro insulin, this is inactive. It is not functional at all. Okay, so how can I convert this into active? Now, one more thing, guys, I'll always, tell you. See, wherever these, hydrogen, I mean, disulfide linkages are present, you need to understand that these are going to be present only at those places where you have cysteine residues. Okay, so here also you're going to see that, oh, there is cysteine. Okay, so here also there was cysteine, and that is why there was this bond formation. Okay, everybody getting this thing? So we have these cysteines, and only at those places we see this disulfide linkages. Okay, but now guys, to convert this insulin into active insulin, we need to do a little bit of work. Okay, and now what is that work, guys? So to convert this into active, now guys, once again I want your entire attention. Okay, to convert it into active ones, we need to remove the C peptide. Okay, so we need to remove the C peptide, and along with that, that NH2 group which was there, we need to remove that as well. Okay, if now we do this thing, okay, if now this is done, now what will happen? See, from here we have removed the entire C peptide. So if the C peptide has been removed, you are going to see that, oh, this was the H in, oh, 1 minute. Okay, so now guys, to convert this into your active one, we need to remove the C peptide. So now if I remove the C peptide, guys, see what will happen. So you have the A chain, the A chain will be as it is, like this. This A chain is now attached to your B chain. So see, the B chain will be like this. Okay, and both of them, how are they going to be linked together, guys? They are going to be linked together with the help of disulfide linkages. So this way, so maybe here also like this, right? So this is going to be active. But guys, one thing that is also going to happen, what you are going to see is, you are going to see that this NH2 group that was there, that NH2 group, this is going to be cleaved off from here. And then you're going to get, this is one, this NH2 group has been removed. You are going to see the second thing that is going to be this active insulin. And plus the third thing that you're going to see is the C peptide. So the C peptide is going to be out over here like this. This is how your actual insulin is going to be done. Okay, everybody getting this thing, guys? Okay, still, but now the next thing that we need to know is, see, if okay, chalo, this is how your normal insulin is made in the body. Now what people did was, if we have to produce this insulin, like, artificially, what can be done? Okay, so the first person to ever talk about this insulin and how this can be done, his name was Edward Schaffer. Okay, so the first person to ever talk about insulin and how it can work, Sir Edward Schaffer. Okay, but now guys, see, he did not prove it though that this can work. So who proved this thing? This was proved by Hakura. Hakura et al. Et al means what, guys? Hakura et al meaning group. Okay, et al means his group, or few other people. Okay, and when did this guy say that this can work, and he proved this thing in 1977. Okay, and now let's try to see what did he do. Okay, chalo, theek hai, fine. Now, see guys, what did he do was, he said it is very simple. Now, what we will do is, we will understand this process and we will try to do it differently. Okay, so what he did was, he said, okay, chalo, I have this B chain. Okay, so the B chain is like this. Okay, so we have B chain. Attached to this B chain, we are going to have this C chain out over here, the C chain, C peptide basically. And then attached to that, we are going to have this A chain. Okay, this is the structure. And then once again, these two are going to be attached like this, like this, right? Now, guys, what did they say? See, anyways, we did not require your C peptide because ultimately that was cleaved, right? So now what did they do? They did a very different thing. They said, we will take the gene which codes for this A chain. Okay, so they said, okay, I will take the gene which codes for A chain. Okay, fine. And now on the other side, guys, they said, okay, on the other hand, I am going to take the gene which codes for B chain. Okay, gene for A chain and gene for B chain. Okay, fine. Because see, ultimately, I had to require only those two. Okay, now what they did was, they did a very different thing. Okay, so now they took a plasmid. Okay, and then what they did was, in that plasmid, they added this gene. Okay, so let me just show you what is that gene going to be. So let's say here we are talking about this gene. Okay, so they remove this this gene. Okay, fine. Okay, now they removed this gene, guys, and then they put this thing in a plasmid. Let's imagine this is the plasmid. And besides the plasmid, like, where they inserted this besides beta galactosidase. Okay, now this has, a marker gene ability. That is why they use this thing, beta galactosidase. Okay, you need to just remember this thing. Okay, fine. Chalo. So we have beta galactosidase, and now you have your, the gene out over here, like this, inside this plasmid now. Okay, now what they did, guys, they just took this plasmid, okay, and now they put this thing in your E. coli. So in the next case, guys, we are going to have this E. coli. Okay, so E. coli has its own DNA, circular DNA, like this, like this, like this. And now, along with this, it is going to get this thing as well. So, chalo, okay. So you are going to have it this way. And now we get like this. Okay, now guys, what did they do? They said, okay, okay, let this E. coli, okay, so we have this E. coli, let this E. coli multiply. So the moment this E. coli multiplies, guys, it is going to give rise to clone. Right? It is going to give rise to large number of clones. And these large number of clones, guys, each one of them, what are they going to produce? They are going to produce your A chain. All of them, they produce A chain. So see, we have got our A chain out over here. Okay. Now the same thing we are going to do for your B chain. So let's imagine now for your B chain, guys, you have the B gene. Okay, the gene for your B chain. It is like this. Okay. Now, what are we going to do? We are going to once again put it in a plasmid, which is going to be like, besides your beta galactosidase. So let's say this is your plasmid. Once again, you have taken your gene, you have inserted your gene out over here. Once again, where besides beta galactosidase. Once again, why are we using beta galactosidase, guys? Because this is going to be a marker gene kind of thing. That is why it helps you to identify which cells have taken it so that you can easily screen them and identify them. Okay. So you have the gene for B chain. It is like this. Now what are we going to do? We are going to take this thing, we are going to put it into your another E. coli. Once again, E. coli is going to have its circular DNA that is out over here. And now once again, what are we going to see? We are going to see that, okay, this entire recombinant DNA, rDNA, it has entered into your E. coli. So once again, what are we going to do? We are going to allow this E. coli to multiply. As this thing multiplies, guys, what is this going to produce? This is now going to produce B chain. Now see, guys, we have got what we wanted. We have multiple number of A chains. We have multiple number of B chains. Now what they did, they brought about a process in a test tube, basically in vitro, by which they added this A chain and B chain. And immediately after addition of A chain and B chain, guys, what did we see? We saw that slowly this gave rise to your actual this gave rise to your actual insulin that is properly functional insulin. But now, how is this? This is going to be synthetically made, meaning it is, laboratory made insulin. And this laboratory made insulin, guys, this is nothing but your human insulin or humulin. Okay, so this is the idea. And guys, while doing this thing, all these reactions, the enzymes that which we have used, this is trypsin. Okay, there is, we will make use of this enzyme called as trypsin. That way. So this is how it is going to be like. So if everybody okay, how we have produced artificial insulin, everyone. Guys, I am not able to see your messages. Yes, guys, anybody has any doubts out over here, guys? Because in your exams, they will always ask you this. Okay, how is this insulin made? What happens in this insulin? Okay. And guys, now just like, if you just to show you how this is done diagrammatically, I have captured this diagram as well. So see the same thing, what I said. See, we have gene for your A chain, we have gene for your B chain. We have put this thing into plasmid separately, put it into your E. coli, allow the E. coli cells to multiply, but they are going to, produce your A chain and B chain respectively. You take these A chain, B chains, and you just mix them together, and immediately we are going to see that, oh, you have got your insulin. Now, guys, Saying this is very simple, but in reality, it is not such a simple task. Okay, this is a very, very excruciating task, actually. Okay, so just have a look at this, guys, to see if everybody is okay. Then we'll move on to the next part. Before these, they used to use dogs or cows. Yes. So how did that happen? That was very simple. So what they used to do, they used to remove the insulin from the blood itself or from the pancreas itself directly. So they used to take some portion of the blood. That blood used to be purified, and from that purified portion of the blood, they used to remove insulin. But then that was your dog or cow insulin, basically meaning cattle insulin. So that was like, from animal. But then what used to happen was, whenever you obtain something from other animals, no matter whatever, how much ever purification you, maybe sometimes, the dog proteins would stay back. So they used to cause, interference. They used to cause allergies, inflammation, when the person used to take administer insulin. So there were issues associated with that. Okay, but then that was directly from the blood or directly from the pancreas. Okay, everybody all right with this? Sure, no issues, guys. Okay, chalo. Now let's try to move ahead. Okay, now after this, guys, we are going to move on to your vaccines. Okay, so chalo, we have produced insulin, fine. Now let's try to go and look at vaccines. Okay, now first of all, what is an what is a vaccine? A vaccine is nothing but it is an antigenic preparation. Okay. Now, what do I mean by antigenic preparation? Guys, it means this is something which is going to elicit an immune response. Oh, okay, elicits an immune response. Now, what does this mean? So, guys, let's imagine. Okay, just a moment. Which elicits an immune response. So, guys, let's say you have your arm. Okay, let's say this is your arm. Okay, now when you take the vaccine, guys, okay, so let's say this is the injection, they have put vaccine into you. Okay, now what is the idea? So basically what we are trying to do in this vaccine is we are actually going to take some killed germs. Okay, either they are killed germs or they are weakened germs, who cannot cause you the disease, because see, you don't want to get the disease while you're getting the vaccine. So for that matter, guys, what we do is, let's say, okay, so for example, we are talking about your BCG. Okay, the BCG, this is bacillus of calmette and urine. This is generally taken for your tuberculosis. Okay, so guys, what I want you to see is, see on your left arm. Okay, on your left arm out over here, there is a little khadda. Okay, I just see this thing on your left arm. Most of you will have a little khadda kind of a thing out over here. I say thoda sa khadda types. Okay, and you must have thought, अरे ये तो मेरा birthmark है, ये तो मेरा birthmark है बचपन से है. Guys, that is not your birthmark. That is your BCG vaccine. Okay, so what happens is when you take this vaccine, you get a nice big pimple, and then that pimple goes away and the khadda stays out over there. Okay, now in our case, in some cases, guys, in some of you, you may not even have this khadda. But in some people, we are going to see small khadda. But if you look at your parents, guys, if you look at your parents, they will have a big khadda. Sometimes you wonder what have they really taken. It's a big khadda. Okay, so huge one like this. Okay, but then anyways, that is nothing but the mark of the vaccine. Okay, but now guys, what are we going to do? Okay, so I told you these are either going to be killed or they are going to be weakened germs, and we are going to put it into your body. Now, when I have put them into your body, guys, see, will they cause the disease? No. Okay. So, these cannot fight. But at the same time, what happens, because they are foreign and they have been introduced into your body, immediately your WBCs. Yes, yes, right. Absolutely. Okay. So you are going to see that these WBCs are going to be activated. And these WBCs, they come, they start fighting with this. But now, guys, what happens? The moment they start fighting with this, guys, these WBCs will always keep a registry, मीनिंग दे विल कीप अ चेक ऑफ अटेंडेंस काइंड ऑफ अ थिंग कि अरे ये है इसका ओके सो ये ऐसा दिखता है ही प्रोड्यूसेस दिस वे ही इज अ थ्रेट फॉर मी एंड दैट registry is going to be maintained. Okay, now what happens, guys? Now these are going to clear off these germs completely, but next time when another germ enters, these WBCs already know this is the culprit. This is going to be someone who's unwanted. As that germ enters into the body, immediately they'll launch an entire immune response and they are going to cut them out and they're going to kill them. That is what is going to happen. And that is how you get immunity to your diseases. Okay. And what is the meaning of elicits and immune response? The same thing, that these WBCs, they're going to keep on fighting. They produce antibodies. They keep on fighting with it so that you are safe that way. Okay. So now, guys, when we talk about this antigenic preparation, this is vaccine. But now, what are the different types of vaccines? Let's try to see. So we have an entire set. Okay, so the most common ones we have your killed vaccines. Okay, so the killed germs. Yes, absolutely right, Sia. Okay, so we are having killed germs. This is one variety. Okay, second thing, we have living but weakened germs. Okay, like half murdered germs who cannot cause any diseases. Okay. Okay, living weakened germs. Okay, now living weakened germs, I'm sure you must have heard of this term also, attenuated. Okay, sometimes you don't understand what is this term, but living weakened germs are actually called as attenuated germs, meaning whose the power of causing the disease has been removed off. Okay, then guys, third category we have living virulent germs. Oh, living virulent germs, meaning in order to get the disease you will put actual germ inside your body. No, guys, this is how actually vaccines were discovered, but we no longer use this method because this is really deadly. Because in this case, in order to get rid of some disease or provide immunity, you first have to suffer from that disease. And guys, some diseases, they are deadly. So you cannot just take that deadly germ, put into your body, you suffer from it, and then you become immune. No, we are not going to do that. So this method, it has been stopped long ago. Okay. And lastly, we have one more method of producing vaccines, and that is called as toxoids. Now, what do we do in toxoids, guys? See, for example, sometimes, okay, the germ itself is not going to be bad, okay, but the toxin is going to be bad. So for example, when we talk about clostridium is not really deadly, but the clostridium toxin that it produces, maybe it is the botulinum toxin, the tetany toxin, the tetanus toxin, that is going to be really bad. For example, we have diphtheria. Okay, so corynebacterium diphtheria. Corynebacterium diphtheriae, it produces a toxin, and that diphtheria toxin is the one which causes the problem. So the germ is not the problem, but the toxin is a problem. In those cases where the toxin is the problem, guys, we go for this kind of formulation, which is called as toxoid. So what do we do, guys? In this case, we take the toxin from the organism and now we mix it with something called as formalin. Okay, formalin, formaldehyde. So that way. And when we do this thing, guys, toxin plus formalin, this is going to be your toxoid. Okay, but now what is the importance? See, this toxoid can be administered into your body. And now there are two things. First thing, this is going to remove the toxicity of the toxin. So this preparation becomes completely safe for your body. So removes toxicity of the toxin, rendering it harmless. Okay, so the moment it removes the toxicity, this becomes harmless, and now you can take this preparation into your body. Okay, and second thing that is does is, even when you add formalin to this, guys, what you will see is the ability to be recognized is not going to be hampered. Your body will still recognize this toxin and it will still produce antibodies, antitoxin against it. Okay, okay, so here the toxin can still be recognized by WBCs. Okay, by WBCs or by your body this way. So these are the two-way benefits of using toxoids. Okay, everybody all right with this thing, guys? Right? So this is what we need to know with regards to vaccines. Okay, nothing much in detail. But now, how has biotechnology developed? And what are the different things that we are seeing? So, guys, as biotechnology has developed, what we see is it has come up with various rapid diagnostic kits. Okay, rapid diagnostic kits. Now, what are these? Okay. So guys, just to give you an idea, okay, rapid diagnostic kits. See, guys, you must have heard of Prega Sure, Prega News, right? Those are those strips kind of a thing, right? And these days now you have got Covishield, Covi, Sure, so, something like the new thing that has come up. कौन है वो? Akshay Kumar is like promoting it. Okay. No. They're not diabetes strips. Diabetes strips are different. Okay. Let's understand. I'm showing it to you. No. We're not talking about we're not talking about vaccines. Okay. Uh, घर बैठ के टेस्ट करो in 20 minutes. Yes. That is what I'm talking about. Yes. That is something which is new, which has come up. I don't really recollect the name. It is something COVID test, COVID sure, something of that sort. That test has come up. Covi self, Covi self. Very good, Himanshu. Covi self, yes. Ha. So now what happens is, guys, see this rapid diagnostics kits. What are we going to do? You are going to take your sample. Okay, so let's say in this case it is your nasal sample, or let's say it is your urine sample. Okay, I'll tell you about another one also. So if you have seen the ad, guys, in that ad also they explain, like when you do this thing, when you take your sputum or when you take your nasal thing and you put it onto this thing, like you have a little buffer, you put some drops out over here. If you get one red line, only one red line, it means the kit is working properly, meaning it is showing some reaction, it is working properly, but you are not suffering from the disease. But then, if by any chance you get two red lines, what does this mean? It means that even you are actually, positive. So you are suffering from the disease. What is this? This is nothing but rapid diagnostic kits. Same way, guys, we use rapid diagnostic kits for your uh, pregnancy also. So we have Prega Sure, Prega News. Okay, what do you do in that case? Once again, the same thing. You are going to take your urine, and now you take a few drops of urine, you put it down on this thing. Now, when urine, see, basically, what are we testing out of it? Oh, let me ask you that. So, guys, in this urine testing, which hormone are we looking for, guys? Does anybody remember? You have done this thing in reproduction. Anyone? Absolutely right. Very nice. Okay, you are looking for your HCG, human chorionic gonadotropin. Okay, we are looking for that. And what is this? So your urine has HCG, and this strip, it has actually anti-HCG. Okay, so antibodies against your HCG. Yes, right. Viva is also right. Huh. These are not 100% accurate. No. So what you do is, see, you always need to do it twice or thrice. Okay, guys, just a moment. Ah, so the thing is, guys, what you need to do is you always need to do it twice or thrice. And how it works is, like when you do it twice or thrice, every time you're not going to get a false positive result. So that, completely removes of that doubt. Okay, so you cannot base your this thing. At least you need to do minimum 2 or minimum 3. Actually, generally, we as scientists we use minimum 3. Okay, that is why. Okay. So now let's say you have taken your urine and you have put your urine on top of this thing, guys. What we see once again, if the kit is working properly, if the kit works properly, you are going to see one line. Okay, and then what does this mean? If you get only one line on the strip, that means you are not pregnant. But then, if by any chance you get two lines like this, guys, now what is this? This means you are pregnant. Now, whether this is a good news or whether this is a bad news, that depends upon you. So that way, but this is nothing but your rapid diagnostic tests. Okay. Anyways, so this way. But now, what have we done, guys? We have done these rapid. See, we are talking about your vaccines and all. So basically, we have done rapid diagnostic kits for various kinds of your bacteria. Okay. So, for example, we have done this thing for salmonella, shigella. Okay. Now, guys, I'm just giving you this entire group: E. coli, coliforms. Guys, why do we do rapid diagnostic tests for this? Guys, because yes. Okay. So why do we do this thing? Because, guys, this entire group of organisms that I have listed out over here, this is because if by any chance someone has defecated into your water or into your water body, these organisms will come into this. Okay, so how it is basically, salmonella contamination, this is going to result into typhoid. Shigella will result into shigellosis. E. coli will once again give you, like, problems, intestinal stuff. Once again, the coliforms, E. coli is actually a part of coliforms. These are a huge group. But once again, the same thing, guys, it is going to affect you and it is going to lead you to diseases. So that is why we check this thing. Okay, so always remember in your exams, okay, in your exams, they ask you, okay, what is an indication that your drinking water has been contaminated? And then they will say presence of certain organisms. Okay, so let's say there is Staphylococcus aureus, then there is another one that is your Bacillus subtilis, then we have Shigella, or then let's say we have your Klebsiella pneumoniae. Okay, and from this four you have to find out. So I'll give you a way, guys. Just remember, okay, there is a water body, okay, and just remember it as, like, in this water body, ह्याच्यात कोणी शी केला? Answer is Shigella. Okay, just remember it that way. You will remember it somehow or the other. Okay, anyway, chalo. So guys, after this, we are going to have the other ones, that is your, this has been done for bacterial ones. Okay, so we have done this thing for bacterial, we have done this thing for your viral stuff. Okay, so all these diseases we have actually, have a rapid diagnostic kits. Okay, but now the next thing that we are going to see in this, guys, is your novel vaccines. Okay, so novel vaccines, these are something that we are trying out right now, and these are like really, really, like we have got, like, really nice responses for this. So unlike your traditional vaccines, okay, what we have done is we have taken naked DNAs, okay, and inserted them into your cells. So we call those kinds of vaccines as naked DNA vaccines. Okay, same way we have viral vector vaccines. Okay, generally viral vectors, we use it to deliver genes and all, but now here we are going to take those the gene for your, like, producing your antibodies or something, and that is nothing but your viral vector vaccine. Okay, and the other one is plant derived vaccine. So these are actually some novel vaccines that we have come across. Okay, so but now, guys, next one. After this, in your novel vaccines only, okay, now we have many other things like we have something that is called as oral vaccines. Now, why will we go for oral vaccines? Okay, let's understand. Okay, understand this thing, guys. First, we will go for oral vaccines only in those cases. Okay, cases, oral vaccines. Okay, only in those cases, guys, where administration is not possible. Okay, like injection is not possible, or when the injections are going to be not stable. But what we have always seen is we will mostly go for your oral thing. For example, guys, if yes, absolutely right, Komal, I just want to talk about polio vaccine. So if you have seen polio vaccine, hi sala, do, hi. Okay, so how it is that? Okay, sir, why meningococcal vaccine is to be taken for students going for abroad studies? See how it works now. There are going to be a set of immunizations which are prescribed. Because why? You don't want you take one disease from here and you take it out over there, and then you start spreading it out over there. See how your COVID has spread. You don't want that thing to happen. So you need to have an entire set of vaccines. It is not just one. There are like 7, 8 vaccines which you need to take before you move from here to any other country. And every country has its own listings. So you need to check for that way. Okay. So now, guys, see, why are these oral vaccines taken? Okay. Oral vaccines, these are done because they are easy to administer. Okay. So easy to administer. So that will why this thing. Second thing is, guys, these are easy to store as well. You don't want to spend extra, अरे, ये क्या हो गया? ठीक है. Okay, you don't want to spend extra amounts of money and all on on its storage and maintaining cold storage facilities and all. So if I can make it in such a way that it remains stable, then I am going to go for your oral vaccine. Otherwise, you'll have to get it injected into your body. But if you have ever seen, guys, you'll always, notice that those injections and all vials, especially, they need to be stored under refrigeration. So for that, once again, your refrigeration costs, they are going to come, electricity costs, they are going to come. So all those things gets added onto it. Whereas in most of the oral cases or oral vaccines or even tablets for that matter, guys, see, we don't have to store them in refrigerator. You just keep it in your normal cool places away from your sunlight. So that is why this oral vaccine is important. But guys, when we cannot use this oral vaccine, when if for any reason the vaccine is not stable in your stomach. Because see, what is going to happen when you take the vaccine, it is first going to enter into the stomach. From the stomach, it is going to go into your intestines, and from there it is going to be absorbed, right? Or something is going to happen out over there. But if your vaccine is not stable in the stomach pH, is there any use of taking oral vaccine? No. Then that vaccine is going to be broken down in your stomach itself, guys. So the first and the foremost criteria is that the vaccine should be stable in your acidic pH of the stomach. Then only it can be classified as oral vaccine. Otherwise, we cannot use it. Okay. Just like we have oral vaccines, guys, we have edible vaccines also, on the other hand. Okay, edible vaccines. Okay, let's try to understand what are these. Okay, so it is basically a category of your oral vaccines only. Okay, so now, guys, what happens? Let's try to see. Okay, just a minute. Okay, so now, guys, you have to understand this entire process. Okay, see, the entire idea is edible vaccines. Okay, so something is going to happen. Okay, chalo. So let's try to see. So, guys, let's say we have a bacteria. Okay, let's say this is some random bacteria. Acha, oh, okay, okay. ठीक है, I'll give you a 5 minutes break. Okay, guys, quickly, 5 minutes break. Then we'll continue with this concept, guys. Okay, 5 minutes break quickly. It is reaching 8:25. Okay, by 8:30 we'll once again start. Okay, quickly. Okay, chalo. Let's move ahead, guys. Okay. So now the second next thing is like, how are we going to have these edible vaccines? Okay, so let's try to see this thing. Okay, so now guys, what happens is some of the bacteria, okay, they are going to produce certain antigens. Okay, so now what happens is, see, let's try to see. So this bacteria is going to have its own DNA, something like this, right? Now this DNA, guys, basically is going to have a gene. Okay, so let's say this is the gene that we are talking about. And this gene is actually responsible for producing some kind of your antigen on the top. Okay, so this antigen, it is going to help me recognize this bacteria. That is how the ability, I mean, that is how the antigen is going to work. Okay, so let me just put down, see, this is your antigen. Okay, now, and if you want, I'll just mention what is this. This is going to help to recognize the bacteria. Okay, and one thing that is like, when are we going to go for this edible vaccine method, guys? Only and only when this antigen, it needs to be stable. Okay, when this is going to be stable when given orally. Otherwise, this will not work. Okay, when given orally. Okay, now guys, in this case, what are we going to do? See, we are interested in this gene. So what we do in the next case, guys, we are going to remove that gene off. Okay, because we only want that gene to be there. Okay, so see, the gene was out over here, something like this. So guys, once again, what are we going to do? We remove this gene off. And now on the other hand, guys, we are going to take the TI plasmid. So we take the TI plasmid. Okay, so this is our TI plasmid. Okay, on the top, it had this tDNA, if you remember. So we had this tDNA. Okay, now what are we going to do? We are just going to open it up, okay, by using your restriction endonucleases. And now we are going to add both of these together. So the moment we have added this, now guys, we are going to get your recombinant DNA. But now this time the recombinant DNA is going to get the gene of your interest. Okay, the one who is going to help us identify the bacteria. Okay, now what do we do, guys? This TI plasmid, okay, now this is, but then this is a recombinant DNA basically. But this TI plasmid, now we are going to put it into your this thing, into your cell. Okay, so let's say this is going to go into the cell like this. Okay. Okay, so now we have put this thing, like, after the agrobacterium tumefaciens has infected the cell, now you are going to see a situation like this. Okay, so you are going to see that this is the plant cell, and this TI plasmid, it has entered into it. Okay, but now guys, just see, if it has entered into this, now what is going to happen inside this cell? Isn't it going to produce that antigen? Yes, it is going to produce an antigen. So what will happen is these cells will be loaded with those antigens like this. So it will keep on producing. Okay, fine. Now let's try to understand how is the repercussion of this thing. What really is going to happen? So when it produces this, guys, now let's zoom out and let's look at a plant. So you will see that, oh, inside this plant, it is going to have the leaves and you're going to have like this. Okay, but now here with each and every cell out over here, guys, what is it doing? It is producing those antigens rapidly inside its cell, like this, like this. Okay, and these are going to be present everywhere, not only in this part. Okay, this is going to be present everywhere. But now what, guys? Now let's try to see. Okay, now let's imagine you are a pan eater. Okay, you are a pan eater. Now what happens? You take this leaf and you're going to make a pan. Going to make a pan of it. Now, when you make a pan of it, guys, you are going to eat the leaf. Okay, but now the moment you eat the leaf, what is going to happen? It is going to go into the stomach. But in the stomach, what is this going to be? It is going to be stable, right? So it is now going to be stable. But now after this, guys, what is going to happen? This is going to be absorbed into the blood. Okay. But now, if it is absorbed into blood, what is going to be absorbed, guys? This is nothing but the antigen, because along with this thing, along with the leaf, you're also eating onto the antigen. You're chewing the antigen, you're breaking it down. So the antigen gets absorbed into the blood. But once the antigen gets absorbed into the blood, what is going to happen, guys? Your WBCs immediately they're going to recognize. What is your body thinking? Your body will think, "Arey, ye to normal food product hai." Right? Because while absorption, they should just think, "Arey, this is just a protein. This is just this thing. I need to absorb it." But we did not know that, oh, this is going to be something different. So immediately the WBCs, they get activated. And now as they are going to be activated, guys, you will see that the WBCs, they start producing antibodies. Okay, now they may start producing antibodies or they may show you phagocytosis also. That depends. Okay. And now, because of this, immediately you are going to receive immunity and you are going to be immune to the disease this way. Everybody got this thing? So these are some novel techniques that have come up. Okay, just have a look at this. Just see if you're okay with this part, guys. Okay, so the idea out over here is when you eat the plant, immediately you are going to be immune to that disease. That is the idea. Is everybody okay? Has everyone understood, guys? AB is antibodies. WBCs, they produce antibodies or they show you phagocytosis. That depends on what response you're going to get. That way. Okay, everybody all right with this? Sure, no issues, guys. Has everybody understood? Okay, chalo, fine. Okay, let's try to move on to the next one, guys. The next one that we have melt in the mouth vaccines. Okay, now these kinds of drugs, basically, they have been available since a long time, but now this is the same kind of technique we're using for vaccines now. Okay, so let's try to see what is this melt in the mouth vaccines. So the principle remains the same. Okay, so generally what we do this thing is this is done for your flu vaccines, and we are also going to do this thing for your bacillus. Okay, so okay, so basically what are we going to do? See, guys, in this case we are going to take the vaccine. Okay, and now this vaccine is going to be put under the tongue. Okay, now, why are we going to put it under the tongue? Guys, because under your tongue, you are going to have an extensive capillary network. If by any chance, guys, any of your parents is suffering from heart issues and, they are prone to heart attack and all, they will always keep a small tablet with them. Okay. And it has been advised that the moment you feel a little uneasy, you take that tablet and put it under the tongue. What happens? Immediately the tablet disperses and all that, the drug, whatever that is there, immediately it gets absorbed into the blood rapidly, and then it is going to be circulated into the blood. Okay, so that is why we will put this thing under the tongue. Once again, what is the reason? Because you have a huge network, extremely huge network of blood capillaries. Okay, and because of this, immediately the vaccine dissolves out over there, and this is going to be rapidly absorbed. Yes, Neha, go on. Okay, that's it, guys. That is what we need to know with regards to your this thing. Okay, but now the next thing is with help of your biotechnology. Okay, how have we changed few things and how have we made use of them in agriculture? Okay, so this was in your medicines. Now let's try to see in agriculture how have we made use of them. Okay, so biotech. Okay, so guys, the most important thing that we have done is we have made transgenic plants. Okay, now transgenic plants. Now, guys, whenever we use the term transgene or transgenic, what does it mean? It means that your genetic material has been modified. So we have made transgenic plants. And what transgenic plants are we talking about? Guys, we are talking about BT cotton. We are talking about BT brinjal. You see, they are not real ones. They are not the original ones. We have changed their this thing. Okay, so we have BT ladyfinger. Okay, etc., etc., etc. Now we have genetically modified them, and that is why they are transgenic plants. But guys, why will we go for this transgenic plants? There are many reasons. Okay, there are many benefits also. So we have first thing that it provides is pest resistance. So they offer greater resistance to any kind of damage. Sir, how can vaccine be given in the mouth? No, 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 no. We are going to take, let's say that vaccine is in the form of some drop or in the form of some tablet. And you just need to put it under your tongue. That's it. Immediately it is going to be given. See, the idea will remain something like this only. See, that it contains the antigen will enter into your mouth. In from your mouth it is going to enter into your blood. From the blood it is now going to be it is going to be acted upon by your WBCs. So that way. So that okay. So we have pest resistance, we have your insect resistance, we have drought resistance, guys. Okay, so we have drought resistance, we have salinity resistance. Okay, we have okay the salt resistance, we have uh apart from that, we have environmental resistance. Okay, so all these different kinds of resistance we can include in your plant, okay, by using your biotechnology and by making use of certain genes, which you want to be expressed in your plant. Okay, next thing is, guys, we have just like this, we have transgenic animals. Okay, now why will we go for transgenic animals? The reason is because they are going to give us maybe high amounts of protein. So their protein, like meaning their muscle mass is going to be more. For example, let's say we produce transgenic chicken who are going to be really, really huge. That means it is going to increase their meat value. But then at the same time, is it good or is it not good? That is a question of debate. That is a different thing altogether. But I'm just giving you an idea. So this is going to help in increase in protein production. So we have this cow called as Rosie. Okay, so what we did was we changed her genetic material, and then what happened was she started producing milk, which was completely laden with proteins, huge amounts of proteins. So that way, but once again, whether to consume or whether not to consume, guys, that is a different case altogether. Okay, generally we don't promote GMOs because we don't know what effect it would have in the near future. Okay, the testing is incomplete. Okay, then we are going to use it to produce clones like your dolly, the sheep. Okay, so we have different kinds of clones we can make. Okay, we can also use this, transgenic animals to test vaccines, or to test some drugs. So generally, if you are into your testing and all, guys, we use some special line of your organisms. And when you use that, they are sometimes very, very susceptible, or they are like easily able to detect cancerous chemicals and all. So that is why we make use of this transgenic animals. And as the most important thing, guys, this is actually used for your experimentation. Okay, and mostly 99% of the times, what are we going to use? We are going to use transgenic mice. Okay. Why is human cloning banned? Because it is unethical. Because see, what may happen is tomorrow you may give rise to a clone of you. Okay. That clone may be like, let's say that is put in prison. You are just allowing it to grow. Why? So that tomorrow, let's say your kidney fails. What will you do? You will take his kidney. Let's say tomorrow your heart is failing. You will take his heart. So that way, so it is not ethical at this moment. Okay. So it's a huge thing. But then what I'm giving you is just a small idea. Okay, now the next thing that we're going to talk about is tissue culture technique. Okay, yes, okay. So let's try to see in this tissue culture technique. Now, guys, why will we go for this tissue culture? It is basically a part of your biotechnology only. Why are we going to go for this tissue culture technique, guys? Because by using tissue culture, we can save. Okay, this has saved those plants which were on the verge of extinction. Okay, because what have we said, guys? If you want to do your tissue culture experiment, you just need to remove only the top portion, apical meristem, and with that apical meristem, you can convert that into like hundreds and hundreds of plants in your laboratory. So it has saved those plants also, okay, which were on the verge of extinction. Okay, so first thing, second thing, guys, this has made us come up with season free plant. So we can sometimes now get plants throughout the entire year because now we are no longer dependent upon the seasons. So for example, guys, if we start developing these uh so also there is a threat of making 100. Yes, absolutely, yes, right, right, yes, true. Okay, so season free plants. Along with this, guys, you know what we can also do by using this tissue culture technique, we can help in or we can bring about conversion of seeds. Meaning, okay, in conversion of seeds, we have something like vernalization. Meaning, I'll just explain you. So you have vernalization or you have yarovization. Yes. Okay, so basically the idea behind this is, guys, you are going to take these seeds and you're going to like subject them to cold. So what happens is because you are subjecting it to cold, suddenly they may change from the winter variety to the summer variety, winter variety to the spring variety. So that keeps on happening. So their characteristics change. In fact, in some cases, seeds which require long day times will be converted into short daytime plants. So long day plants into short day plants. So something of this sort. So all this thing is going to happen. And also, guys, because of this tissue culture technique, what are we also going to use it for? To increase the production of plants also. Okay, and by increasing the production of plants, what are we going to do? We are going to increase your food production. So for all these purposes, we are going to use tissue culture techniques. Okay, fine. But now, next one, we have transgenic plants. Okay, so transgenic plants, we can also use them. Okay, so transgenic plants, we can make them with higher nutritional values. Okay, so for example, your golden rice. Okay, so with increased nutritional values. And now when we use this, what is this going to do? This is going to tackle malnutrition. Okay, so malnutrition in children, malnutrition in the masses, whatever you like. Okay, so this is what we need to know with regards to your entire transgenic plants and all. But now the next thing, guys, we have gene therapy. Okay, so let's try to see what is this gene therapy and how does it this work. Okay, so guys, gene therapy means what? Okay, so as the name suggests, you are going to introduce a gene somehow or the other, and by that, you're going to give some therapy. So basically, guys, in this gene therapy, the genes which are defective, okay, the genes which are defective, non-functional, non-working, guys, we are going to treat them. And this entire process of treating them is nothing but your gene therapy. Okay. But now, how are we going to do this thing? What are we going to do? So, guys, either you are going to replace the gene, okay, or you are going to alter the gene, or you are going to supplement the genes. Okay, so by doing this, we are going to, try to give gene therapy. But now, in most cases, okay, like most diseases, like when we talk about most of the diseases, they are going to be genetically related. Okay. And now, in order to get rid of this, guys, okay, so first, let me give you a little idea which are the diseases. So, for example, guys, once again, we have hemophilia, we have sickle cell anemia, right? So, let's say we have muscular dystrophy. Okay, let's say we have cystic fibrosis, right? So, so all these are like your genetically, genetic diseases like this. And now, in order to overcome these diseases, guys, we are going to make use of gene therapy. So wherever these defective genes are present, we are going to replace them, okay, either in their place itself, or we are going to use some supplement, genes, meaning you take the proper gene, you try to somehow introduce it into the cell so that now it starts functioning that way. So that is how we will make use of it. And then, guys, next thing, we have single gene defects. Okay, and to treat this thing, I mean, basically we're going to use once again your gene therapy. So in your single gene defects, who are we going to have? Once again, sickle cell anemia, we're going to have thalassemia. Okay, we have Tay-Sachs disease. Okay, Huntington's disease. Okay, we have hemophilia, alkanopturia, etc. etc. So all these are nothing but these are certain genes which we can this thing we can we can treat. But now, how will we do this thing? Okay, so first I'll just put this thing down. Next class we'll see exactly how the situation is going to be. Okay, but first let me just introduce you what are the different possible options that we have. Okay, so I have this thing. So see how we can do this thing. Either replace somehow or the other. Okay, somehow or the other, you replace the missing or the defective gene. Okay, or second thing, you take the deliver gene, deliver the genes that are going to destroy your cancer cells. Okay, maybe you can take those genes and you can put it into cancer cells, which is going to make them come back to normal cells once again. Okay, deliver bacterial or viral genes in the form of vaccination. Okay, deliver DNA to antigen expression and generation of immune response. So whatever we have spoken, like how the antigen is going to be taken up into the body and how is it going to be expressed that way. Supply of gene for impairing viral replication. So here you're going to stop. Right? Sickle cell anemia. SCA is sickle cell anemia. Okay, so here in supply of gene for impairing viral replication, here you are going to stop the viral the viral virion from multiplying. That is how it is going to be. Okay, provide genes that promote or impede growth of new tissues, and lastly, deliver genes that stimulate healing of damaged tissues. So these are different ways by which we can, introduce genes and we can bring about, betterment, basically. Okay, so guys, this is what we need to know. The next time, okay, next time when we come back, guys, we will talk about something that is called as virosomes. Then we will talk about how is gene therapy going to be like. So in gene therapy also, we have two gene therapies. We have germline therapy, we have somatic cell therapy. Okay, and then the next thing that will be is how are we going to deliver the genes into the cells? Okay, so we are saying that, okay, the gene is going to be put into the cell, but how is it going to be done? That we will see it in the next class, guys. Okay, chalo. So guys, once again, we'll stop over here for the day and I'll see you in the next class. Till then, take care. Bye bye, everybody. Bye bye.